DNA replication follows this process as shown in an example of bacterial DNA replication, in which the helicase unwinds the parental double helix first. This is the helicase. And then molecules of single-strand binding proteins stabilize the unwound template strands. So the leading strand is synthesized continuously in the 5 to 3 direction by DNA polymerase 3. The lagging strand also synthesizes from the 5 to 3 direction, so it synthesizes backwards. And with the primase begins the synthesis of the RNA primer for the 5th Okazaki fragment. This is the primase. So DNA polymerase 3, also this red molecule here, is completing the synthesis of fragment 4, seen here. When it reaches the RNA primer on fragment 3, it will detach and begin adding DNA nucleotides to the 3 end of fragment 5 primer in the replication fork. DNA polymerase 1 removes the primer from the 5 end of fragment 2, replacing it with DNA nucleotides added one by one to the 3 end of fragment 3. After the last addition, the backbone is left with a free 3 end. So, and then this DNA ligase joins the 3 end of fragment 2 to the 5 end of fragment 1. And then remember that this is the lagging strand. DNA is composed of nucleotides, which consist of a backbone made of deoxyribose sugar and a phosphate group and a nucleotide base. The nucleotide bases include adenine, thymine, guanine, and cytosine.
Thymine and cytosine are pyrimidines, and adenine and guanine are purines.
Nucleotide pair substitution. This type of mutation is a change in one DNA base pair that results in the substitution of one amino acid for another in the protein made by a gene. Insertions are additions of nucleotide pairs in a gene. Insertion of nucleotides may alter the reading frame of the genetic message, the triplet grouping of nucleotides on the mRNA that is read during translation. Such a mutation can lead to a frameshift mutation. Deletions are losses of nucleotide pairs in the genes. Like insertion, it can cause frameshift mutations.
For more specific DNA mutations, there is point mutations, which changes a single nucleotide, silent mutations, which do have a mutation in one of the nucleotides, but it doesn't affect the phenotype of the protein that is assembled. Missense mutations change one amino acid into another, but the amino acids are similar enough that there is little effect. In nonsense mutations, the amino acid is changed to a stop codon entirely, stopping synthesis of the protein.